Hello everyone, and thank you for watching another video on qualitative and quantitative analysis of pharmaceutical excipients. Today I'm going to talk about cetostearyl alcohol. This video is useful for people who are doing reverse engineering of pharmaceutical or cosmetic products. I appreciate if you could write a comment about your experience with cetostearyl alcohol determination, or any comment that you find useful for everybody. Well, cetostearyl alcohol is a mixture of cetyl alcohol and stearyl alcohol. So depending on the raw material that you buy, this ratio could be different. It is extensively used in pharmaceutical and also in cosmetic products. It is a fatty alcohol, and the function is more like a stabilizer. It will stabilize the cream formulation, and it will prevent it from separation, or probably it will use for the viscosity booster or other functions, but primarily it's used for stabilizing cream products. There is a monograph on cetostearyl alcohol in USP, and it describes that it is actually a mixture of the cetyl and stearyl alcohol, and cetyl or stearyl alcohol content cannot be less than 40%, and overall sum of the both fatty acid cannot be less than 90. So again, depending on what you buy, you may have different ratio of these two fatty alcohol, and in your reverse engineering you may find that maybe this ratio is different in the raw material that you have in house versus what you see in the product that you are reverse engineering. Also in EP there is a monograph for cetostearyl alcohol, and it very much describes the same thing as USP says. A stearyl alcohol minimum it cannot be less than 40%, and overall sum not less than 90%. So it's a compendial raw material. In FDA IAD database, cetostearyl alcohol mentioned number of times. 7 entries for topical drugs, 3 entries for oral, 3 entries for vaginal. The topicals, we have cream, gel, lotion, ointment, spray, suspension, with the allowable limit of between 1% in spray up to 12% in cream. In oral, we have the extended release, using extended release tablets and capsules, and the limits are 9.34 milligram or up to 62 milligram, depending on the dosage form. In vaginal products, we have 12% weight per weight. FDA IAD database also have limits for cetyl alcohol and stearyl alcohol. I don't want to go through them. These, maybe you can look at it yourself. But overall, the allowable limit for individual cetyl and stearyl alcohol appears to be less than cetostearyl alcohol. So probably this is the reason that we see cetostearyl alcohol in the formulation a lot more than individual cetyl alcohol or the stearyl alcohol, because the allowable limit for the cetostearyl is higher than cetyl and stearyl individually. For qualitative and quantitative analysis, I'm just going to talk about GC and HPLC LSD detector. For the GC, USP monograph has the assay method that describes how you do quantitation of cetostearyl. As I said before, this method is aimed for raw material testing and not for reverse engineering. However, you could use this with some modification. The issue with the USP method is that, in my opinion, is using the packed column rather than a capillary column. And people who do GC, they know that the packed columns are not very popular anymore, and everything is capillary column. So that's an issue that I probably have with this method. On the other hand, if you look at the EP monograph, it has an assay method, VGC, but they are actually using the capillary column, 30 meter, 0.32 nanometer. It's a very, and it is very suitable column for this work. And the stationary phase, it is a DB1 or ZB1 column. So if there is a choice between either using EP method or USB, I definitely would recommend the EP method. Now this is the chromatogram. Is not entirely EP method. I use DB5 instead of DB1, and the solvent is heptane, and the internal standard is a fatty alcohol. You could use a fatty alcohol. And in this chromatogram, we see the two peaks of cetyl alcohol and stearyl alcohol around 5 to 6 minutes, and the other peaks are the other excipient present in the formulation. This GC for cetostearyl alcohol determination is very robust, and I rarely use anything other than GC for quantitation. So the two peaks that we see here, you can also look at the ratio of these two alcohols and make the quantitative determination. That is getting information about the ratio of these two fatty alcohol. Then you could compare this with the raw material that you have in house, and you will pick a proper raw material for your reverse engineering work. So with the GC you can get amount determination, and also you can do some grade determination, both. Now with the HPLC ELSD you could do the same thing. This is the chromatogram that we're looking at here is that a formulation that contains a lot of fatty material, surfactant, and cetostearyl alcohol. The two peaks at 6 minutes and 8 minutes are peak for the cetyl and the stearyl alcohol, that they are well separated. The column is a C8 column, and the mobile phase is a combination of water, acetyl nitrite, TFA, and ethanol. I would say the ELSC method development, you will be more challenged. It is more challenging than GC, and if I want to make recommendation, I would say GC is for sure. And for some particular reason, if you choose to do HPLC ELSC, yes, you have a choice as well. So the references, I would just make a reference to USB NF monograph and EP monograph, and probably there are many other references that they talk about cetostearyl alcohol determination. Thank you very much for watching this video, and have a great time.